So welcome everybody to today's Ento Live. The focus today will be on the honeybee and how the National Honey Monitoring Scheme is looking at honey to help us understand what long-term changes in the health of our countryside are occurring. So I'm going to hand straight over to Jenny so that we can get on with what I'm confident is going to be an absolutely fascinating talk. So over to you, Jenny.
Brilliant. Thank you very much, Kieran. Thank you for the opportunity to speak today on Ento Live, and really appreciate you all still coming along. I know it's a gorgeously sunny day out there, and so I'm honoured that you're still tuning in to listen to me talk. Normally, if we were in person, I'd ask for a little hands up in the room on who's heard of the National Honey Monitoring Scheme before and who might have taken part. But I don't think I can actually see your reactions if we do that now. So I'm going to give this talk on the basis that no one's heard of it before or participated. If you have, then hopefully there'll still be quite a lot in here that you don't know, because it's a sort of behind the scenes of the honey monitoring talk. So if you did want to follow us, our Twitter is ch_honey. I'm Jennifer Shelton. I took over running the scheme in October last year, but a project like this is never the work of a single person. So you can see here everyone that is involved in this project. It was the brainchild of the PI, Richard Pywell. It was set up by Anna Oliver in 2018, and she did an absolutely amazing job of running it until I took over last year. And I just want to give a big shout out to Ellie Grove, who does a lot of this behind the scenes lab work as well, and to our funders listed at the bottom. So just in case you were expecting me to be Ben Woodcock, it wasn't available to give a talk today. I do have a few of his slides at the end of my talk as a little teaser, and hopefully Kieran can get Ben in to talk about the pesticides at a later date. Okay, so who are we? Who runs the National Honey Monitoring Scheme? We're the UK Centre for Ecology and Hydrology. We have about 500 scientists across 4 UK sites: Edinburgh, Lancaster, Bangor and Wallingford, which is where I am in Oxfordshire. We're the UK Centre for Excellence in Environmental Science across water, land and air. And we generate the data and understanding that researchers, businesses and governments need to create a productive, resilient and healthy environment. What is the National Honey Monitoring Scheme? So it's a UK-wide citizen science project asking beekeepers to send us honey samples for us to analyse. So we asked them to send in 3 30 ml honey samples collected from their hives. One is for pollen analysis. And this is the pipeline that I'm going to be focusing on today, because this is the one that I am in charge of. And we use that to monitor the impact of land use change and agriculture on honeybee foraging behaviours. We asked for a second tube, which is for pesticide analysis, which is what Ben would have been talking about, which is to measure the exposure of honeybees to pesticides whilst they're foraging. And then we asked for a third, which goes into our honey archive. And this is a repository of all of the samples we've ever analysed for future research projects. And we believe that NHMS is the largest environmental DNA citizen science project in the world. So the background to NHMS, as I mentioned, it was set up in 2018. It was established in consultation with the BBKA and the Bee Farmers Association. When I sent out a newsletter last week to everyone who's ever participated, we're actually up to 3,330 subscribers to the newsletter, and they're from all over the UK, England, Wales, Scotland and Northern Ireland. We've so far published a few papers about it. One is our pipeline that you can see here, bottom left, which enables anyone to do the analysis pipeline that we're doing on honey, to make that open access and an open resource, and an initial presentation of the results, which has come from this citizen science monitoring using honeybees. So why did the scheme choose to work with honeybees? It's because they are so widely kept in the UK. We have about 46,000 beekeepers who registered as of 2021 on BeeBase. In the hive count of February 22, there was 272,631 hives registered. And this gives us a huge pool of beekeepers who are really enthusiastic and willing to help with this research. So honeybees are essentially living environmental monitors. Everything that they are exposed to within their 2-kilometre radius of the hive, they bring back to the hive and shows up in the honey as they process that. And the honey itself is quite inert and it lasts for a very long time. So it's a way of preserving these exposures in a way that we can analyze them. This is an overview of the pollen analysis pipeline, and I'm going to talk to you about this in more detail. Please don't be worried if you're not a scientist by background. There is a little bit of sort of genetics and molecular biology in here. I'm going to try and explain it as well as I can, but if you do have any questions, please pop them in the chat as we go along or scribble them down and ask me at the end, and I'll do my best to explain things in a different way. So to start off with, we ask beekeepers to collect the honey samples directly from the comb in their hives. They send it to us at UKCEH. We filter the pollen grains out of the honey, and then we extract the DNA from those pollen grains. We then amplify a plant-specific gene, which is known as a DNA barcode, and then we sequence it on a next-generation sequencing platform. We get back millions of reads for what plant that pollen belongs to, and then we use bioinformatics to match those reads back to the samples that they came from. And then we send out the results to the beekeepers. So when a beekeeper logs into our website, they need to put in the location of their hive and how long their hive has been at this location. And then we post out a sample pack to the address that they give us. The sample pack looks like this. So it'll have a brief explanation of what we'd like them to do, why we'd like them to do that, free post-return envelope, 3 sample collection tubes, and a plastic bag. So we always ask them to put the honey samples in the plastic bag because honey, as you know, is mega sticky. And if there is a leakage, which there very often is, it's very nice that it's contained within that plastic bag. So we ask that the honey is collected from the same frame from a single hive and that they're all collected at the same time from capped honey. And this is because we want to see a snapshot in time in what's happening in that single hive. Whereas if they were to send in extracted honey samples, that extracted honey could come from multiple hives, possibly from multiple apiaries and across a long duration of time. So it's not quite what we're looking for because NHMS is foremost a scientific study. And that's what we're looking for, is to know when the honey was collected. We ask them to ensure that the location is accurate because when you request a sample pack, you may put in a location of a different hive that doesn't end up being as productive. And then you collect your honey samples from a hive elsewhere that has turned out to be more productive. And we just ask everyone to check that their information is correct for the hive they've sampled from, and this can be updated at any time. So this, I am actually a beekeeper myself, as it said in the intro notes. This is a crazy picture of me looking quite warm in May 2018. Pure coincidence, I started beekeeping the same year that this scheme started, and this was my first super of honey, so that's why I look so chuffed. So this is the form that we also send out online when you sign up to get a sampling pack. The only bit of information that we ask is what date the sample was actually collected, but we really do encourage beekeepers to fill in the rest of the form because it lets us know what type of hive the bees are in, how long you've had that hive, how much honey you expect to get from the hive, and some questions about the health of the hive, whether the beekeeper's seen any signs of disease or had any queen losses. We can get some really interesting notes from beekeepers about this, and it just feeds into the science that the analysis that we can do with the data. So this is a picture of our honey archive room. I mentioned that we take that third tube and we store it in the archive. So the left-hand side of the picture is all of our archived samples since 2018. And the right-hand side, this is how the honey archive room looked when I started last October. So that is the backlog of all the 2022 samples that were ready for processing. And there was 1,500 honey samples there waiting for me when I started. So the first step of the pollen analysis pipeline is to take that one sample that has been designated for pollen analysis. We weigh out 15 grams into a fresh tube, and then we also put some honey on a refractometer, and this measures its sugar and water content. And that's the first piece of data that the beekeeper receives, is the sugar water content on their sample. And I've just updated each stage of this pipeline with a rough timing, just so you can get an idea of how long it takes to process a honey sample. So this stage, the refractometer and the weighing out into the tubes, we can do about 10 samples per hour. So the next step is to filter the pollen grains out of the honey. So to do this, we top that 15 grams of honey up to 50 mils with molecular grade water. We warm it for an hour at 55 degrees with regular mixing, and this makes sure that all the honey and the wax within the honey is suspended in the water, and that all of the pollen grains are also loose within the water. We scoop out the wax. Wax is a good sign because it shows us that it has actually been collected straight from the comb as we requested and is not extracted honey. So we scoop that out, and then we put that honey and water mixture into a vacuum cup. And at the base of this vacuum cup, I'll just show you with my mouse, is a piece of filter paper here. So once this cup is filled up with honey, we apply a vacuum pressure to the cup, and that draws that honey through the filter paper and then gets discarded. And then you can see here on the right, this is the clean piece of filter paper, and this is what it looks like after the pollen grains have been filtered. So this is interesting because depending on how much is in the honey, you can get a lovely range of colours on the filter paper, from this sort of pale yellow all the way through to a really deep orange or brown. We can do about 48 samples per day at this processing step, the filtering stage. So at this point, we have those pollen grains sat on top of filter paper, ready for the next stage. And I found these tweets by Dr. Jacqueline Gill, a paleoecologist at the University of Maine. And I think she does a really good job of summarizing how tough pollen is. And that explains why it's so tricky to do the next step. Pollen is made of sporopollenin, the most resilient natural biopolymer known to science. You can pull out pollen grains from hard rock dating back tens of thousands of years. Pollen is an amazing forensic tool, helping us to reconstruct not just crime scenes today, but to tell the stories of the planet's ecosystems and civilizations in the past. And those stories provide clues to climate future. Did I mention how resilient pollen is? You can douse it with concentrated sulfuric acid and hydrofluoric acid, the stuff that dissolves sand, and it doesn't even care. To close, pollen is a versatile tool in paleoecology and archaeology. Pollen is beautiful, resilient, and powerful. It's also super cool. It's the plant equivalent of identifying a giraffe from a lizard using only sperm over thousands of millions of years. So that brings us to the next stage, is how do we crack open these super resilient pollen grains that have evolved to resist everything the natural environment can throw at them, to get out the DNA that we need for the sequencing step. So we do a process which is called lysing. So we add those pollen grains into a tube, then we add a chemical concoction to that tube. So this breaks down the outer layer of the pollen, and we also include an enzyme in there that breaks down all the proteins in that mixture that would damage the DNA if left to its own devices. Then for those pollen grains that have resisted the chemical lysis step, we put them in what's called a bead beater, and that vibrates the tube incredibly quickly and violently, and mechanically lyses open those pollen grains. So at this point, we have a mixture that contains all of the cell debris, the outer casings of that pollen, all the DNA inside that we want, but also all of those other cell components, which if we left it as that cell lysate, the DNA would quite quickly get degraded. So then we move all of that lysate into these amazing filter tubes here, which have a matrix on the bottom, which binds only the DNA. You put that tube into a centrifuge, and the rest of the lysate gets flushed through the tube, excuse me, but the DNA remains bound to that matrix. You add what's called wash buffers onto the matrix, which just makes sure that it's only DNA that's bound, and it cleans through everything else in those steps. And then once you're confident that you only have the DNA that you're interested in, you add what's called an elution buffer, and that binds to the DNA preferentially over this matrix. And then when you centrifuge that tube, you end up with your DNA in the elution buffer here. And this is what's called a DNA extraction at this point. And we'll have a DNA extraction for each honey sample that has been weighed out and filtered in the previous step. Don't be scared off by this slide. It's a little bit complex. All I wanted to say about this is that essentially, when you go into a supermarket and you scan the barcode on an item, and then the supermarket till recognizes that item, is exactly the same with DNA. Every plant, fungus, insect has a barcode in its DNA that we can essentially isolate and use it to identify that species. So for plants, we're using a barcode called ITS2. And essentially from the last step in that DNA extraction, we have all of the DNA from the plant in that DNA extraction. And we only want to access this small fragment, which is about 500 base pairs, because of the limits of our sequencing ability at this stage. So we want to pull out that 500 base pairs by isolating it with what are called primers. So these are short fragments of DNA that bind either side of the barcode that we're interested in. And then we perform a PCR, which everyone's heard of now, thanks to COVID. And that PCR amplifies the DNA, and we end up with thousands of copies of the barcode that we're interested in versus single copies of the rest of the DNA. So we can focus on the barcode. And then time-wise, we can PCR 96 samples in 3 hours. So for anyone who's done any molecular biology before, you'll be familiar with this slide. This is quite a nerdy slide for me to include because I just find this process particularly fascinating. This is called gel electrophoresis. Essentially, we make a thin gel, essentially, with holes at the top, wells at the top. In that gel, there's a fluorescent stain that binds to DNA. So then when you put the PCR amplicons into the top of the gel, if you have DNA, if the PCR has been successful and the DNA extraction before that, if you have DNA, then this fluorescent stain will bind to it. And then because DNA has an electrical charge, you apply a current to the gel and it carries that DNA through the gel. Smaller fragments move faster than larger fragments. So then you end up with this laddering effect. And then when you shine a UV light onto the gel, that fluorescent stain fluoresces. And if it's bound to DNA, it will show the DNA in the gel. The left-hand sample here is what we load called a DNA ladder. Oh, sorry, didn't mean to click that. And that gives us the different sizes of DNA. So essentially, we can tell the size of the DNA that we've extracted from our samples according to this ladder. We can also say at this point whether our DNA extraction or our PCR has been successful. So for example, these first 4 samples are beautiful ITS bands here in the correct size. We can tell that that's between 400 and 500 base pairs, but you can see that this sample hasn't worked. And this picture on the left here just shows you what it actually looks like when you light it up with that UV light. You can see that fluorescent stain and the DNA. So then the next step, because we can only load a single sample onto the sequencing machine, this is called a MySeq machine, we label all of those sequences with a unique combination of code. So we put tags on the beginning of that DNA barcode and at the end, and each sample has a unique pairing of codes so that we can pool them. We place them on the sequencer. It produces millions of reads for that combination of samples. So we currently pool 394 samples together at this stage. And it takes about a week to do all of the steps from cleaning up the PCR to pooling and to sequencing. And then once we have all of those reads, we can use those unique pairing of tags to tell which samples they came from initially. So once we have those collection of DNA barcodes, we run them against a DNA database. So this just gives an example for a single plant, might have multiple DNA barcodes, and you can see here the ITS2 barcode. So we match our sequence reads against that barcode, and then that tells us what plant the DNA belongs to that was extracted from the pollen. And then we do some taxonomic identification. So this is just an example for Rubus, which are brambles. The kingdom Plantae, or plants, contains about 280,000 species, which then narrows down to division, angiospermae, or flowering plants, which contains 250,000 species. Then you narrow it down to rosales, which is roses and their allies, which is about 18,000 species. Then down to the rose family, which is 3,500 species. And then down to the genus Rubus, or brambles, which is about 250 to 700 species. So depending on how specific your DNA barcode is, it allows you to get down to different levels of certainty within this taxonomic identification. With ITS2, you can reliably get down to genus or species level because of the specificity of those 500 base pairs between different species. Rubus or brambles is slightly different because there's so many subspecies and cultivars and hybrids that are very closely related. That DNA barcode isn't specific enough to be able to tell apart the individual species. So we get down to a Rubus level, but we're unable to tell apart, for example, a blackberry from a raspberry or a loganberry from a tayberry, because that sequence will be too similar within the 500 base pairs. There is a limitation, but for the analysis that we're doing, that ITS2 region is a really good DNA barcode to choose. So another example with brassicas, we normally only get back a brassica result, which is normally oilseed rape. And that is because if you cross turnip or Chinese cabbage, which is Brassica rapa, with Brassica oleracea, you get oilseed rape. However, if you cross Brassica oleracea with Brassica nigra, which is black mustard, you get Ethiopian mustard. And if you cross B rapa with Brassica nigra, you get Brassica juncae, which is brown mustard. So all of these have very similar sequences for ITS2 because they are so closely related. But we assume with honeybees that when we get a brassica result, it's oilseed rape, because that is the crop that they will fly for miles to visit if they have the option. So once we have that data back, we send a bespoke report to each beekeeper, which is specific to their hive. The first data point that I told you about, they get back is the sugar water content of their honey. But then they will also get the land cover in the 2 kilometers surrounding their hive, and they will get this report back on what their honeybees have been feeding on, which really is the thing of interest for beekeepers and the bit that I get the most questions about. So they'll get the top 15 taxa, plant taxa, that have showed up in their honey, and then they'll get the full list of all plant taxa that has showed up. So that land cover is broken down into 2 pie charts. They'll be given one pie chart showing the habitats and crops surrounding their beehive, and one showing the crops and another showing the habitats. And these are calculated from the land cover maps that are produced here at UKCH. And these are produced from satellite images that assign land cover and crop to about 90% accuracy, I believe. And thanks to my colleague Emily Upcott, who does that analysis. So the total sample is about £110 for this entire pollen analysis pipeline. Right, so I'm just going to tell you a bit about the story so far and present some results from 2019. So the scheme's grown massively. When it was set up in 2018, it was more of a trial year. So 202 samples were sent in for analysis. Then the following year, there was 586. In 2020, probably helped by the pandemic and everyone being stuck in lockdown, this jumped up to 1,109 honey samples. Following year, 1,164. And then as I was saying last year, which is when I started, we were up to nearly 1,500 honey samples. And this plot here just shows you where they come from. As you might expect, the majority of the samples come in from the southeast and southwest. This seems to be where the majority of beekeepers set up their hives because of the lovely weather and the fantastic honey flow. So we're always trying to target other areas, more the Midlands and into the north. So this is the sign up for this year so far. I think we've achieved a really good coverage already. However, we are looking for more beekeepers from central Wales and anywhere in Scotland. But we appreciate it is a harder sell for beekeepers the further north you go, because the honey yields go down. So I completely understand why they don't want to scoop out and send us their precious honey. So this is just an example report for 2019. No, I apologise, this is a summary of all of the data that was analysed in 2019. So about a quarter of the pollen in those samples came from brambles. And as you remember from my previous slide, that can be a collection of lots of different raspberry, loganberries, tayberries, blackberries. Then next up was brassicas, most likely oilseed rape, but also a combination of the others. And then sweet chestnut tree and white clover. And then you can see a drop down here from 9% to the 2s and 1% of borage, wild radish, privet, Himalayan balsam, forget-me-nots, and broad beans. So they were the top 10 taxa that bees were foraging on in 2019. And we just put a little red arrow next to this one here for Himalayan balsam, because that's an invasive species. So it's actually really interesting that we can use honey samples to monitor for the spread of invasive species here within the UK, if they're of interest for honeybees to forage on. So the average number of plants per sample was 28. And there are about 10% of plant taxa that were not on UK plant lists. So they're ornamental plants. They're likely bought from garden centres that are not native to the UK. So then you can look at various different metrics. Richness or alpha diversity is the diversity of all of the plant species that are in a particular honey sample. So you can group those honeys together by different metrics. Here they've grouped them by where the region of the UK that they came from, to look at whether there's any difference in the diversity of what the honeybees are feeding on in different regions. And actually looking at that plot, we would say there's really nothing significant there. However, when you group them by the months that the honey was collected, you can see this interesting trend in the diversity of the species that the honeybees were feeding on. So there seems to be a high diversity in April, which you think is when a lot of the fruit trees are flowering. Then a drop off in May, interestingly, and then it builds back up from June to another peak in September and October, and then drops off in November. So different type of plot now. You can break it down by the plant species or the genus that you're finding in the different ones. Again, there's not much to be said about the regions. You can't really pick out a huge number of trends, but I can just highlight a few of the most common plants that are detected. So this grey blue here represents the brassicas. The grey here represents the brambles, and the pale sage green is the woodland forget-me-nots, the Myosotis. So these appear most predominantly across all of those samples. However, when you break down these by month, again, you can see this really nice transition in what the bees are feeding on. So in spring, it starts off with mainly oilseed rape and these woodland forget-me-nots. In the summer, this moves to brambles and clover. And then in the autumn, it moves to Himalayan balsam and ivy. And if any of you are beekeepers, you can also see this change in the honey that you collect off the hive. For me, oilseed rape honey is very pale and it sets, whereas by the end of the season, the ivy honey is much deeper colour. It never sets and it has an entirely different taste. So for me as a beekeeper, it's also very interesting to see these data. So another way of looking at this diversity is to see whether it changes by what is in the land cover surrounding the hive. So here, if you look at increasing agricultural land cover, so this is from 0 to 1% all the way up to 90 to 100% agricultural land cover. What these plots are actually showing is that as the area surrounding a honeybee hive becomes more agriculturally intensive, the diversity of what the bees are feeding on declines, which actually makes sense, because if you're a bee and you're surrounded by oilseed rape in the 2 kilometers surrounding your hive, then the only option you have is to feed on oilseed rape, really. This is another way of showing that. So they've taken 5 hives here that have between 0 to 10% coverage of agricultural land, and then you make connections to all of the different plant species that each hive is feeding on. And you can see that each of these hives have absolutely hundreds of connections to different plant species. And these yellow bands here represent the oilseed rape. And they make up maybe a quarter of what those hives are feeding on. However, then when you increase the agricultural surrounding to between 70 to 90%, all of a sudden that oilseed rape is making up probably 75%, if not more, of that, what the honeybees are feeding on. And there's much fewer connections and much lower diversity of the plant. Okay, so that was a little teaser on what we found so far from the honey data, the pollen analysis. Now I'm going to talk a few slides by Ben Woodcock about the pesticides residues in the honey. So focusing on neonicotinoids, they were first licensed in the UK in 2002 for use on oilseed rape. they very quickly dominated the insecticide market, and they represented 24% of the market by 2008, until they were found to have these sub-lethal effects on bees. So the EU introduced a moratorium in 2014 for mass flowering crops, leading to a full ban in 2018. However, there have been exceptions for sugar beet in 21 and 22. So here you can see when the insecticide market before neonics was dominated by this gamma HCH, but very quickly the neonicotinoids took over. And then by 2010, there was 3 main types in use. So prior to the ban on the usage, this is what the use of clothianidin looked like. This is the use of the thiamethoxam, and this is the use of imidacloprid. So pretty widespread use across the UK. Ben and colleagues did some modelling with the coverage of oilseed rape in UK, also the deployment of neonics and other pesticides. And they overlaid these with volunteer surveys by Biological Record Centre and the Bees, Wasp and Ant Recording Society of various different bee species. And then this is an example for Adrena chrysosceles. The blue line here shows that if neonicotinoids were not having any impact on this bee species, this is what you would expect to see on these bee recording surveys. However, this was the actual trend shown in red. So you can see this decline that has been attributed to neonics. Different way of showing it here. Those bee species that are not predominantly foraging on oilseed rape show a much less of a reduction in their population than those who are foraging on oilseed rape. So clothianidin was still used on wheat, and it was still detected in the honey residues for years after the moratorium. So whereas imidacloprid, please excuse my pronunciations, was increasingly phased out prior to 2014 and has hardly been used since, and you can see this decline in its residues. So less than 20% of neonicotinoid seed treatments are actually absorbed by the crops, which means that the rest of it is free to be absorbed by other plants surrounding the crops. So on this plot here on the right, you can plot the predominant plant species that appears in honey with pesticide residues in. You can see that the highest concentration of neonicotinoids appears in those honeys that have the highest content of oilseed rape. However, those medium concentrations of neonics appear in plant species that are predominantly planted in the field borders around oilseed rape fields, and then the same for a lot of wildflowers that are found in verges surrounding farmland. So they assessed 82 fungicides, herbicides and insecticides in the honey samples that were tested. There were at least 2 pesticides in each honey sample. One had 17, and the most pesticide residues were found in May samples, and even urban honey was contaminated. There are residues found in areas with 0 agricultural surrounding the hive. Even pesticides that were not approved for use in 2019 were found in the honey. So for fungicides, of 13 no longer approved, 6 were still detected in at least 1 honey sample. For herbicides, of 8 no longer approved, 4 were still detected. And insecticides, of 14 no longer approved, 13 were still detected. So this could be a combination of farmers having stock of those pesticides in their barns and deciding they'll use it up until it's gone, or more likely it's an indicator of the persistence of those residues. So even though we can ban something and stop its use, it doesn't mean that it just disappears from our environment, and therefore it's still having this effect on our pollinators as they go about their daily activities. Okay, so thank you very much. That was a bit of a whistle-stop tour through the pollen analysis and a few teasers on the pesticide analysis. I really hope that that all made sense, but please feel free to ask me more questions, particularly about the molecular genetics side, if that made no sense at all, and I'll do my best to explain. And a very big thank you again to everyone who's been involved in the various aspects of this project, and to our funders. And if you'd like to know more, please visit our website. Thanks very much.